So now we're are talking about cell junctions. Let's get to it. This is chapter 4. First, we have the gap junction, and the reason why I remember that this is a gap, or one thing I did to make myself remember that this is a gap junction, is that they have these little connectons, as you see here. And I think about braces, and when I think about braces, I think about teeth. And I think about teeth, I think about gaps. So if the gap junction has these little bracelets-looking things on it, and they're called connectins. And as you can see on the model demonstrated, that will be located right here. Now, one function, because we have to know their functions as well of a gap junction. They act as a channel for communication, which allows ions and molecules to pass through from cell to cell, and even it helps to communicate nutrition, I believe, as well. Next, we have the hemidesmosome. The hemidesmosome, of course, has a cousin, and I like to say that his cousin is a desmosome. So let's go back to the hemidesmosome. This one is made up of a glycoprotein called integrin. And it also, when you look at it on the model, to me, it looks like a little hot dog. If I can get the camera to focus so you can see, it looks like a little hot dog with some chili and stuff on top of it. So for me to remember that, the glycoprotein that's used in the hemidesmosome, I think about how we have to have integrity to eat a hot dog, okay? Crazy sentence, but it makes sense to me. And we see these little stringy straw things. These blue things are interment filament, which is a keratin. And we only have one side of that. And we also have a basement. And which is, if you get the picture to identify it on a test, you know that if one has the basement, you know that is particularly, it goes there. It's like kind of like a matching game. You know that it would go there. And so one function of the hemidesmosome is that they anchor cells together. They are, I don't want to say they're like what could I say? They connect cells to extracellular material, such as the basement membrane. Okay. Next, we have the desmosomes. So the desmosomes, as you see, is located right over here. They call this particular junction the spot wells, I guess because it doesn't stitch all the way around. Like if you were hemming something, it doesn't stitch. That means you don't stitch all the way around. You just stitch a little, I guess like here, 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 and here. You don't stitch the whole entire thing. And I guess that's like what the desmosome is. And as you can see, there's these straw-looking things again. There's these little keratin filaments, just as the hemidesmosome had, but we have two this time. And also this particular glycoprotein that it contains is cadherin. And it's not the only junction with cadherin. We'll talk about that in a second. But one function of a desmosome is that it holds things together. Well, I'm going to read the description. It says it's composed of plaque and it's linked by transmembrane glycoproteins, as we just talked about. It extends across a gap between the adjunction cell membrane and linked to the cytoskeleton of cells together. So not a deep description but from the book, but basically it keeps cells from being pulled apart to left or right. That's what I've got from another source. And it's found in spots that we have a lot of like stress, like the skin or the intestines that get used pretty often. So desmosomes, they keep cells from being pulled apart. They're composed of plaque. They're linked together by transmembrane glycoproteins. And that's pretty much it for this one. The Adherence Junction. As you can see from the picture, it's located right here. And once again, we want to play a matching again, because look at this blue belt that you see on the side of this junction. You see it also in the model, so you know that this is, of course, the belt. Not only does it look like one, but it actually is a function of this particular junction. So that it's nicknamed a belt because it joins cells together. The book says it's made of plaque and anchors cells together. It transmembrane glycoprotein called cadherin, which is the same protein that's in the desmosomes as we read earlier. So it holds cells together, and it's called the belt. Okay, great. Now, the last one we have is the tight junctions. Now, if you take a look at this tight junction, you see the details. To me, it looks like some little Mardi Gras beads all over the junction. So, it's the bead. Now, just think about holding the bead tight, around your neck. It'll be tight. If it's too tight around your neck, it's tight. So, tight junction, Mardi Gras beads. You get the picture. You get the correlations, and it's located right here. And if you think about it, when we're playing matching again, if you look to the side of this photo, you see some little bumpy looking things, if you can get to focus. So that is a tight junction. It's pretty much like you're playing matching. It's a tight junction here. Yeah, so it's like you're playing matching. This is so fun, right? So one function for the tight junction, and also it adapts the name of the seal or the Ziploc bag because that's exactly what it does. It seals things. It forms tight seals between cells and are commonly among the epithelial cells that line the stomach, the intestines, the urinary, and the bladder, etc. So that is where we will find the tight junction in areas where it's leak-proof. And that's pretty much what it does. It's a leak-proof seal. That's what it is, I meant to say. You keep materials from leaking out of the organs, okay? So that's what it does. And so, with that being said, I want to go back to desmosomes. Here we go. Yeah, I feel like I didn't give a thorough explanation. Okay, so desmosomes, from the PowerPoint, it says that it used the cadherin glycoprotein plus the intimate filaments to hook to the cytoplasma. So that's something extra about this one. Okay, so that's pretty much it, guys. Just review. You do good on the test.